Hello students, today we are going to discuss about next step of a meiosis, that is a meiosis 2. After meiosis 1, if we are going to discuss about meiosis 2, then we can say that this meiosis 2 is also comprised of 4 phases, and that is prophase 2, metaphase 2, anaphase 2 and telophase 2. And also we can say that it resembles the mitosis, because as per mitosis here also during meiosis 2, chromosome number will not halve. Okay, means chromosome number will remain constant. Let me initiate with the prophase 2. What happened during this prophase 2? So first of all, it is initiated immediately after the cytokinesis. As I already told you in previous lecture that during this process maybe cytokinesis occur, may not occur. So there is a two chances. Cytokinesis may occur, may not occur. So if it is occur, then the prophase 2 will be initiated immediately after the cytokinesis. The chromosomes again become compact. Okay, then nuclear loss, nuclear membrane will be disappear in both the nuclei. This is the characteristic feature of any prophase 2, that chromosome become compact, nuclear loss, nuclear membrane will be disappear. As chromosome become compact, then central will be moved towards the opposite end. Okay, and the nuclear loss and nuclear envelope disappear. This is the feature by using which you can say that this prophase 2 is completed and the initiation of metaphase 2 will occur. So now this chromosome will be naked. Okay, chromosome is not covered with the nuclear membrane now because it is disappeared. Okay, as a result of that, this centriole will now form the spindle fiber, and now spindle fiber will be come in contact directly with the chromosomes. So as you know very well, on the chromosome there is a centromere. On the centromere on the both side there is a kinetochore. So this spindle fiber will now attach with the kinetochore. Okay, and as a result of both side it will be attached, and that's why what happen, all the chromosome will be pushed. Okay, here it will be pushed towards the center. From here also it will be pushed toward the center. From here also it will be pushed toward the center. As a result of that, all the chromosome will be aligned in the center on the metaphase plate. Okay, and the chromosome align at the equator, and the microtubule form opposite poles, and that is a spindle fiber of the spindle, and get attached to the kinetochore of a sister chromatid. So each sister chromatid will be attached with the spindle fiber from the both direction, and that's why all these spindle will be push this chromosome in the center. From here also force will be applied, and from here also force will be applied. So due to pushing force, all chromosome will be aligned in the center on the equatorial plate, and that is an indication of metaphase 2. And as all chromosome aligned on the center, this phase will be completed, and that is an indication of initiation of anaphase 2. So during anaphase 2, what happen? These spindle fiber which is attached to the kinetochore will be shrink. Okay, and it will be pull the chromosome. Here pushing process is there, and here pulling process will be there. Okay, and due to pulling of a chromosome, what happen? From the both side pulling will occur. This will pull from here, this will pull from here, and that's why pressure will be generated. As a result of that, the centromere will be break down from the center. Okay, and as a result of that, one sister chromatid will be moved towards this end, and second sister chromatid which is dissociated will be moved towards this end. Okay, so by this way both the sister chromatid will be separated. So by this way both the sister chromatid will be separated from each other from the centromere region, and it begin with the simultaneous splitting of the centromere of the chromosome which was holding sister chromatid together, and thus they move towards the opposite poles of the cell. So by this way we can say metaphase 2 is the result of a pushing method. Okay, here spindle fiber will push the chromosome, and here spindle fiber will be pull the chromosome. So pushing and pulling method is responsible for these both phases. After the completion of anaphase 2, next step will be initiated, and that is a telophase 2. Okay, during which two group of chromosome 1 again get enclosed by the nuclear envelope. As I already told you, the telophase phase is a phase in which reversal will be occur. That is exactly reversal of a prophase. Okay, so during prophase whatever is removed or disappear will be reappear in this case. Okay, so during prophase, what is already disappear? That is a nuclear envelope, nucleolus. So here in this phase, all that which is a disappear during the prophase will be reappear. So here nuclear envelope will be reappear. Then cytokinesis will follow, the resulting in the formation of tetrads. Okay, so 4 haploid daughter cell will be obtained as a result of a cytokinesis. So here compulsory cytokinesis will occur, and as a result of that 4 cell will be obtained. That is a 4 haploid daughter cells. Here in this slide you can see overall steps of a meiosis. So let me summarize that. So first of all, prophase 1 will be followed by metaphase 1. Okay, so prophase 1, you know very well, this is the longest phase. Okay, during which lactotin phase, zygotin phase, pectine, okay, then diplotin and dikinesis. This is the longest phase of a meiosis, and after that it is be followed by the metaphase 1. Okay, and then anaphase 1, then telophase 1. So this is the phase during which chromosome number will be half. So this step is very important. Here only chromosome number will be half in the case of anaphase 1. And finally it will be followed by telophase 1, then prophase 2, okay, metaphase 2, and further in during anaphase 2, centromere, from the centromere will be background, and as a result of that sister chromatid will be separated out, and as a result of that you will get the haploid number of chromosome during the telophase 2. So these are the overall steps. So first of all, parent cell, that is a diploid cell, okay, will undergoes interphase, then prophase 1, leptotin, zygotin, pectine, then diplotin and dikinesis, then metaphase 1, then anaphase 1, telophase 1. As a result of that you will see this is the haploid. Okay, this is the diploid cell. Parent cell will be diploid. Okay, and during meiosis 1, ending of the meiosis 1, you will see a haploid cells. So here you will see during prophase 1, these two cell, parent cell will be already haploid. Okay, so we can say that this is the exactly what happened during the mitosis. So meiosis 1 resemble exactly mitosis. Okay, because here initial cell will be haploid and the resultant cell will also have haploid structure. Whereas in this case, this is the diploid and this is the haploid. So this is the difference between meiosis 1 and meiosis 2. And the reason that why this is meiosis 2, and it is also considered as a resemblance of the mitosis. So prophase 2, metaphase 2, anaphase 2, and finally you will get the telophase. During the telophase 2 you will get 4 haploid cell. Okay, 4 daughter cell you will obtain. Now let me discuss what is the significance of meiosis. So it will conserve the chromosome number of each species. Okay, due to the meiosis process only, chromosome number will be maintained in constant. It causes genetical variation, you know very well. During a pectine stage there is a crossing over occur, and at that time gene exchange will occur, and as a result of that a genetic variation will be obtained, and due to crossing over during the pectine phase in the population of organisms. So person to person we are looking different, organism to organism we are looking different. Okay, so all the varieties of the biological varieties on the earth is just due to this crossing over. So all the plants are different from each other, all the animals are different from each other, even our brother and sister are different from each other, even twins are different from each other. That is all just due to crossing over step. Okay, it is important for the evolution also, and due to this only evolution will occur. So this is a basic step for the evolution. So crossing over is responsible for evolution also. This is your homework for today's lecture, and before we finish this lecture, let me summarize all topic by using one animation video. So look at this video.
Meiosis consists of two cell divisions, namely meiosis 1 and 2. Meiosis 1 is divided into 4 stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Similarly, meiosis 2 is divided into 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis 1 results in 2 haploid daughter cells due to the separation of the homologous chromosomes in the parent cell. Meiosis 2 follows meiosis 1 and results in 4 haploid cells. Meiosis II resembles mitosis, as the same number of chromosomes is retained in the daughter cells. Between the two meiotic stages, there is a short-lived resting stage known as interkinesis, during which the DNA doesn't replicate. Interkinesis is followed by prophase II, a stage that is much simpler than prophase I. During this phase, the chromosomes get thicker, shorter, and distinct, with each chromosome consisting of 2 chromatids. By the end of this phase, the nucleolus disappears and the nuclear envelope disintegrates. Prophase II is followed by metaphase II. During this phase, the chromosomes move towards the equator and align themselves along the equatorial plane. Bipolar spindles are formed, and the microtubules from the opposite poles of each spindle attach themselves to the kinetochores of the sister chromatids. The next phase of meiosis II is anaphase II. During early anaphase, the centromere of each chromosome divides longitudinally into 2 and splits the chromosome into 2 daughter chromosomes. These daughter chromosomes are then pulled to the opposite poles of the cell during the late anaphase stage. The final phase of meiosis II is telophase II. During this phase, the chromosomes reach the opposite poles and a new nucleus with a nuclear membrane is organized at each pole. Did you know that all 4 nuclei differ from each other in genetic aspects due to the crossing over in prophase I? Telophase II is generally followed by cytokinesis, during which the cytoplasm in each cell is equally divided, resulting in the formation of 4 haploid daughter cells. Meiosis is a significant process in sexually reproducing organisms. Meiosis in germ cells, followed by the fertilization of gametes, restores the chromosome number in the offspring of sexually reproducing organisms. Another significant feature of meiosis is that it leads to variation in offspring due to the crossing over in prophase I, during which genetic materials recombine. Variation plays a very important role in the process of organic evolution. Meiosis plays a vital role in sexually reproducing organisms. The process takes place only in reproductive cells or germ cells and is also known as reduction division, as the number of chromosomes is reduced to exactly half in the daughter cells.
In next lecture, we will learn some extra topics for NEET, and that include modification of mitosis and amitosis. So till that, goodbye.